Hi, class, and welcome to Chapter 23 on cellular metabolism. This is divided into two parts, Part A and Part B. I'm just going to be going over Part A. This is primarily what you'll be tested on is in Part A. I included Part B in Canvas because there's a lot of good, interesting information in it. But I'm going to only focus on Part A in this chapter. There's also a great workshop on cellular metabolism respiration. So if you can get to that workshop, that would hopefully help you with some of this material as well. So cellular respiration and metabolism, first let's talk about the words anabolism and catabolism. So anabolism is the synthesis of large molecules from small ones. And an example is synthesizing or making proteins from amino acids. Catabolism then, in comparison or contrasting to that, is the hydrolysis of complex structures into simpler ones. So for example, breaking down proteins into amino acids. So we have these two opposite ideas: anabolism, synthesizing larger molecules from small ones, and catabolism, breaking them down into smaller pieces. Cellular respiration then is the catabolic breakdown of food fuels, whereby energy from food is captured to form ATP into cells. And the goal of cellular respiration is to trap chemical energy in ATP. So energy can also be stored in glycogen and fats, which can be broken down later. The word phosphorylation has to do with enzymes shifting high energy phosphate groups of ATP to other molecules. So phosphorylated molecules will then become activated to perform a cellular function. There's three stages in processing nutrients. Stage one is the digestion, absorption, and transport to the tissues, which we talked a little bit about in our digestive system PowerPoint. Stage two is the cellular processing in the cytoplasm, where we synthesize lipids, proteins, and glycogen, or we catabolize them, or glycolysis. We split apart glucose into pyruvic acid and acetyl-CoA. So this all happens in the cytoplasm of the cell. And then stage three occurs in the mitochondria, which is the oxidative breakdown of these intermediates, whether that's pyruvic acid or acetyl-CoA, into carbon dioxide, water, and ATP. Cellular respiration consists of glycolysis of stage two and all of stage three, and that's what we will focus on. So here are the three stages of metabolism of energy containing nutrients. Stage one occurs in the GI tract, where nutrients are digested into absorbable units. They're absorbed into the blood and transported to the tissues. So proteins are broken down into amino acids, carbohydrates are broken down into glucose and other sugars, and fats are broken down into glycerol and fatty acids. Stage two occurs in the tissue cells. Anabolism, nutrients are built into macromolecules, but in catabolism, nutrients are partially broken down, and glycolysis is the major catabolic pathway that ends with pyruvate. So here we have glycolysis, which takes glucose and splits it apart into pyruvic acid, which then turns into acetyl-CoA. And then in stage three, we head into the mitochondria, which is the oxidative breakdown of stage two products. Carbon dioxide will be released, the hydrogen atoms are removed and are ultimately delivered to molecular oxygen to form water. Some of the energy released is used to form ATP, and then the citric acid cycle and oxidative phosphorylation are the major pathways. And we will focus on kind of glycolysis and stage three in cellular metabolism. We need to understand what we talk about, what an oxidative reaction is. An oxidation reaction involves the gain of oxygen or a loss of hydrogen atoms and their electron. Oxidation reduction reactions are also called redox reactions, where oxidized substances are losing electrons and energy, and reduced substances are gaining electrons and energy. And redox reactions are catalyzed by enzymes that usually require a B vitamin coenzyme. Dehydrogenases are enzymes that catalyze the removal of hydrogen atoms, and oxidases are enzymes that catalyze the transfer of oxygen. There's two important coenzymes that act as a hydrogen or electron acceptor. nicotinamide adenine dinucleotide, NAD+, or flavine adenine dinucleotide, FAD. And these are just really important coenzymes that will act as hydrogen or electron acceptors in the oxidative pathways. And we'll see those a lot, NAD+ and FAD. So here's a look at, for example, turning succinic acid into fumaric acid. So succinic acid is reduced, and we are going to oxidize it, meaning we're going to take these two hydrogen atoms away. And when that happens, we're turning FAD into FADH2, and that's because it will be reduced by taking these two hydrogen atoms. ATP synthesis are two mechanisms. Two mechanisms are used to make ATP from captured energy that is liberated during cellular respiration. Substrate level phosphorylation is when high energy phosphate groups are directly transferred from phosphorylated substrates to ADP. This occurs twice in glycolysis and once in the Krebs cycle, where the necessary enzymes are in the cytosol for glycolysis and in the mitochondria for the Krebs cycle. So this is a look at the mechanisms of phosphorylation. A high energy phosphate group is transferred directly from a substrate to ADP to form ADP. And this occurs in the cytosol and the mitochondrial matrix. Oxidative phosphorylation then is a more complex process, but produces the most ATP. We can have a chemoosmotic process, which couples the movement of substances across the membrane to chemical reactions. The energy released from oxidation of food is used to pump hydrogen across an inner mitochondrial membrane, creating a steep hydrogen concentration gradient. And as hydrogen flows back through a protein called ATP synthase, energy from the flow is used to phosphorylate ADP. This is a look at oxidative phosphorylation as a part of the electron carrier transport mechanism. The electron transport proteins pump protons, creating a high proton gradient above here. And then ATP synthase uses the energy from the proton gradient to bind phosphate groups to ATP. And this occurs only in the mitochondrial matrix. Carbohydrate metabolism occurs when glucose enters a cell. It's phosphorylated to glucose 6-phosphate. Most cells lack the enzymes for the reverse reaction, so glucose becomes trapped inside the cell, and only cells in your intestine, kidney, and liver can reverse the reaction and release the glucose. This keeps your intracellular glucose concentration low, which ensures continued glucose entry. Glucose is catabolized via the following reaction. Glucose plus 6O2 turns into oxygen, water, carbon dioxide, and heat, as well as ATP. The complete glucose catabolism requires these three pathways, which is what we're going to go over and which is what I want you guys to know. Glycolysis, the Krebs cycle, and then the electron transport chain and oxidative phosphorylation. And this is a great summary slide showing where all these processes occur. So during cellular respiration, ATP is formed in the cytosol and in the mitochondria. Glycolysis splits glucose into pyruvic acid. Pyruvic acid enters the citric acid cycle, which then forms intermediates, which will enter the electron transport chain. So let's first talk about glycolysis, and this is the oxidation of glucose. Glycolysis is also called the glycolytic pathway. It involves 10 steps. It can occur anaerobically in the absence of oxygen, but this glycolysis occurs in the cytosol, where glucose will be broken down into two pyruvic acid molecules. There's three major steps of glycolysis: sugar activation, sugar cleavage, and then sugar oxidation and ATP formation. I'll let you go through those three steps on your own. But the big thing that I want you to know is that at the end of glycolysis, we're turning glucose into pyruvic acid. And I'll show that right here. So here is the three major steps of glycolysis. And this third step is forming two pyruvic acids at the end of glycolysis, which will then go on to enter the citric acid cycle. Glycolysis, you have to keep in mind in glycolysis that although 4 total ATPs are made, the 2 ATPs needed to start the phase 1 must be subtracted. So only a net gain of 2 ATP occurs in glycolysis. We also create 2 NADH. So for glycolysis to continue, more NAD must be present to accept more hydrogen atoms. All right, so that's glycolysis. And again, the big thing about glycolysis is I want you to know that it creates 2 pyruvic acid at the end of glycolysis. Then we go to the citric acid cycle. This is also called the Krebs cycle. This occurs in the mitochondrial matrix, and it's fueled by the pyruvic acid from glucose breakdown and fatty acids from fat breakdown. Pyruvic acid is actively transported into the mitochondria because it is a charged molecule. And once inside the mitochondria, that pyruvic acid enters what we call a transitional phase. The transitional phase is where each pyruvic acid is converted to acetyl coenzyme A or acetyl CoA in 3 steps. Those are the 3 steps, but all I want you to know is that in the citric acid cycle, the pyruvic acid first needs to be converted to acetyl CoA. So here's a simplified version of the citric acid cycle. We are taking the pyruvic acid and turning it into acetyl CoA, and that's the first transitional step of the citric acid cycle. The citric acid cycle then uses coenzyme A to shuttle its acetic acid to an enzyme in the citric acid cycle that joins to form with a 4-carbon oxaloacetic acid, which produces a 6-carbon citric acid. And this is why it's called the citric acid cycle. The big thing about the citric acid cycle is that during its 8 steps, the original acetic acid is decarboxylated and oxidized to keto acid intermediates, resulting in NADH, FADH2, and carbon dioxide. And in the final step, oxaloacetic acid is regenerated. So that's kind of the simplified version of the citric acid cycle. For each turn of the cycle, we'll get 2 carbon dioxide, 4 molecules of reduced enzymes, which are the NADH plus H plus and the FADH2, and then 1 ATP. We started with 2 pyruvic acid molecules, so the amount of the final products from 1 glucose is always doubled. And adding the products of the transitional phase, the total final products for breakdown of 1 glucose up until this point is shown here. And again, this is the citric acid cycle. The citric acid cycle, NADH must be oxidized to ensure there's plenty of NAD. So glycolysis, the transitional phase in the citric acid cycle, can continue. And this is a great animation of the citric acid cycle that you can watch when you download the PowerPoint. Then we'll get to oxidation of glucose continuing in the electron transport chain and oxidative phosphorylation. Oxidative phosphorylation consists of 2 phases. Phase 1 is the electron transport chain, which creates a proton gradient across the mitochondrial membrane using the high energy electrons removed from H from the food fuels, and chemiosmosis, which uses the energy of the proton gradient to synthesize ATP. So here's a look at oxidative phosphorylation. Again, it has 2 phases. Phase 1 is the electron transport chain, which creates the proton gradient, and the chemiosmosis, which uses the energy from that proton gradient to synthesize ATP. And this is a great picture showing the electron transport chain and what happens. So these electron carriers, which are coming from the NADH plus H plus and the FADH2, what they're doing is they're pumping their electrons up to form a proton gradient. So we're trying to form a gradient of all these hydrogen atoms up here in the intermembrane space. These proton carriers, 1, 2, 3, 4, are pumping protons across. Ubiquinome, which is coenzyme Q, and cytochrome C will help shuttle electrons between the larger complexes. And then once we get this proton gradient above, we have chemiosmosis, which uses the proton gradient to synthesize ATP. So what we have here is ATP synthase, which is this complex number 5, harnesses the energy of the proton gradient to synthesize ATP. So as hydrogen atoms flow back across the membrane through ATP synthase, the synthase rotor will spin, causing phosphate groups to attach to ADP, which will form ATP. So that's kind of the generalized summary version of the electron transport chain. So in the electron transport chain, it's the only pathway that uses oxygen. The citric acid cycle will not run if oxygen is not present, but does not directly use oxygen. And an overview is shown here. An overview is that the NADH plus H plus and FADH2, both coming from glycolysis in the Krebs cycle, are delivering their hydrogen atoms from glucose to the electron transport chain proteins. The electron transport chain will then use the energy harnessed in the electrons of the hydrogen to pump those hydrogens across the membrane to create this proton gradient. Some hydrogens will combine with oxygen to produce water. I'm going to let you guys read through the different phases, but each phase explains how we're basically just pumping the protons across the membrane to create this pump. So this is showing how these hydrogen atoms get transferred through the proton carriers. And as that occurs, the energy is being lost. And eventually, these excess hydrogen atoms will combine with oxygen to produce water. So we need oxygen as what we call the final electron or hydrogen acceptor, because oxygen will combine with these hydrogen atoms and electrons to produce water so that the electrons and hydrogens aren't just hanging out in the membrane. So again, this is the summary. We have hydrogens being pumped across the membrane from NADH and FADH2. The hydrogens will then flow back through the ATP synthase to create ATP, and then the excess hydrogen atoms and electrons will combine with oxygen to form water. Chemiosmosis then talks about how these protons will then re-enter the matrix via the complex 5, which is the ATP synthase. And the ATP synthase is like a small motor that drives the addition of an inorganic phosphate to ADP, creating ATP. So this is a look at the motor. Protons flow down their electrochemical gradient through the stationary motor or ATP synthase. Each hydrogen binds to a subunit of the rotor, causing it to change shape, making the rotor spin. The spinning rotor turns the connecting rod, and as the rod spins, it will activate the catalytic sites in the knob that will join a phosphate group to ADP, creating ATP. And this is a really cool look of ATP synthase rotor rings are shown thickly clustered together. They look like little donuts with holes in the middle. And you can kind of see them all here in the mitochondria. They'll all spin to really create all that ATP that's needed by your cells. ATP synthase works like an ion pump in reverse. The proton gradient drives the energy needed for ADP, pyruvic acid, inorganic phosphate, and calcium to cross into the inner mitochondrial membrane. And this is a great animation of that electron transport chain. Cyanide gas, which is a gas used in gas chambers, will disrupt the oxidative phosphorylation by binding to cytochrome oxidase. It will block the electron flow from complex 4 to oxygen. This poison will also uncouple or abolish the proton gradient by making the inner mitochondrial membrane permeable to hydrogen. So although the electron transport chain continues to deliver electrons to oxygen, at a furious pace, oxygen consumption rises, so no ATP is made. So that's what cyanide poisoning does. So here's a summary of ATP production. Energy use at rest averages about 100 kilocalories per hour, which is slightly more than a light bulb uses. It places a huge demand on the mitochondria for energy production. Of the 686 kilocalories in 1 mole of glucose, only about 260 kilocalories are captured in ATP, and the rest is lost as heat. So this translates at about 38% efficiency in the usage of fuel available. If you think about an average machine, machines are only about 10 to 30% efficient in using their food source for fuel, and your mitochondria are a lot more efficient than that. Here's the summary of ATP production itself and the net ATP production produced. And I want to get so the total amount between substrate level phosphorylation and oxidative phosphorylation is equal to about 32 ATPs for one glucose molecule, but energy is always required to move NAD generated into glycolysis into the mitochondria. So a final of ATP produced is usually about 30, but there's still uncertainty about that final total. And this is just a good energy yield during cellular respiration, which you guys can review on your own. We get about 30 ATP typical yield per one glucose molecule. And you can see where each of those ATP are coming from in the three major steps, whether it's glycolysis, citric acid cycle, and the electron transport chain. And this is a good final review of cellular respiration that you guys can watch on your own. This is the end of this chapter. I included part B because it has some interesting information, but part A is what I really want you guys to focus on, this cellular respiration. Thanks for listening. We just have one more chapter to do before the end of the semester, and that will be on the urinary system.